here today. Good morning, Ryan.
Morning, Kelly. Good morning, everyone.
We are your TAC co-chairs. We won't go through introductions as we're on Zoom, but you can click on the participant list, which keeps growing by the minute, or I should say actually by the second. It just literally went from 47 to 51 in like one second. So welcome everyone. I'm always glad to have a lot of participation in the TAC. And remember your tools. If you have questions, you feel free to throw it in the chat. We will be monitoring that. And if you're having any technical difficulties, just, again, just tag the person in the chat so that we can make sure we can, if it's on the host side, we can correct that. So with that, we're going to kick off our first presentation, laboratory findings of saxitoxin and Pyridinium bahamense absorption by biochar. Tufek and Reza and his team from FIT and others will be presenting, is my understanding. He's got the whole team here. So I'll let him introduce his team and take it away.
Can you guys share or see my screen?
Yes.
Okay, perfect. Well, thank you very much for inviting us. It is always excited to show us the results, what we are doing, telling you a story, what we have been learning all along, and then where we are going next. So this is almost, we finished the second year. We have an extension for 1 year due to the Hurricane Helene. So we are talking about biochar and modified biochar for saxitoxin and pyridinium removal from water. I'm Tawfiq Reza. I'm an associate professor in chemical engineering at Florida Tech. I have my co-PIs here, Dr. Spencer Fire, also from Florida Tech, as well as Dr. Vince Lovko from Mote Marine Lab. The two students are deeply involved. One is Katie Ann Chambers, should I say Dr. Katie Ann Chambers. She just graduated last fall. And then my new student, Robert Cheatham, he took over the project. So to welcome everyone and thank you for inviting me to present. So we're going to start with a very basic of harmful algal blooms. As we probably know, some of you might know and some of you don't. So harmful algal blooms, they're usually broadly divided into three groups. One is the group that doesn't release toxins, but it actually populate on the surface of water that blocks sunlight, that would deplete the oxygen. So that's my type 1. And type 2 is the one that is producing toxic species and that is harmful for human as well as our sea life. And the type 3 is it's not producing the toxins, but it is going to clog the gills and other things that would affect the sea life as well. So in our group, we are mainly focusing on type 2 because it is our toxic forming harmful algal bloom. And to give you some examples, so we're talking about type 1, the type 2. The first algae and toxins we started to look into was the microcystin LR and produced by blue-green algae. It's a cyanobacteria. But beside that, there are a couple of other dinoflagellates available here in our state. You probably are familiar with the red tide, which is caused by Kyrenia brevis, and we have our separate project funded by USHAB CTI. We just finished our project funded by the Red Tide Initiative on Kyrenia brevis on biochar as well as modified biochar. But we also looked into pyridinium, and this is the project that is funded by Tampa Bay. So we're going to talk about pyridinium and saxitoxin. to talk a little bit about pyrrhodium. As you see, and the slide actually came from one of the project that is funded by Tampa Bay. I think it was, if I'm not wrong, it was run by FWC, FWRI. So it's a very good project, and I was reading about the project. It's funded by 2020, I guess. It was looking into, you know, how many seats are there in old Tampa Bay, and as well as around Tampa Bay. So if you look into the right side, let me use my pointer. There you go. So if you look into that, so it's a different seasons, and then you see that there are like in the sediment, we would have a lot of cyst forming. And if you look into the life cycle of Pyrrhodium bahamense, we would actually see cyst forming, and then it goes into the vegetation, make more and more cyst. And why do we care about Pyrrhodium? Because Pyrrhodium, it produces saxitoxin, and saxitoxin is a neurotoxin that is often related to the PSP, the paralytic shellfish poisoning. And if you look into the chemical structure, as I show it over here, let me just hide the plotting. And you see it is a polar non-volatile. It's a tricyclic parhydropurine alkaloid. It's a mouthful, but this is what it is over here. It looked like when human and the fishes they ingest it, they usually start feeling tingling and burning around the mouth, tongue, and face, and then it would started to do the paresthesia, changing, going through the numbness. So that's why it's called the paralytic shellfish poisoning. Now, we looked into this one, and then my other project looking into different harmful algal blooms, for example, Carenia. One of the major technology that has been, very close to a field demonstration is a clay flocculation. And Dr. Lovko, Dr. Don Anderson, they're running that as well. So one of the things that I have learned from that study is it's very good at flocculating the cells, but it doesn't have the mean to remove the saxitoxin from water. As a chemical engineer, I do work with porous carbon material, and I look into the chemical structure, and I see that there are a bunch of functionality in terms of like OH bonds. We have aromatic rings. We have nitrogen hanging in there. So it could be a functionality. It is a small molecule, relatively small molecule. So maybe we can actually use adsorption as a mean to remove it from water. So that's where it comes in. And then my expertise comes right after using the biochar. So if you are not familiar with biochar, it is not as high as activated carbon. So if you're comparing activated carbon to biochar, the price would be about 1/10 of the price that it is commercially produced. You can buy it from Amazon as well. It is predominantly used for soil amendment. What it does is basically you have your biomass, for example, pine or westwood, and then you actually cook it at really high temperature under inert atmosphere to give you like more stable porous carbon material, as you see in the second slide or second the middle picture. This is our picture taken by Katie, and this is a commercial biochar. You can see a very porous nature. A couple of good things about biochar is it is coming from renewable resources. It is relatively cheaper. We did use it and reactivate it, so it is some form of reusable. But in this project, we are not reusing it because it's going to be collecting it would be a little too expensive for this project. And again, it could be engineerable, so it's basically what means that I can change the processing condition to some extent to fill the need for that. And it is also efficient, we're going to see. So we bring biochar, and then we went to the Tampa Bay Estuary Program, and we come up with three objectives. So first objective is basically I was looking into like, okay, how can we use biochar for pyrrhodinium adsorption, saxitoxin adsorption? The first question is, where do we get our pyrrhodinium? I reached out to NOAA as well as FWC, FWRI, and then it's basically they're only able to give us a 50 milliliter starter kit. Which is very good. And however, for our benchtop study as well as a column study, we need a little bit larger volume. So we said that, and I reached out to Dr. Lovko from Mote Marine Lab. He has tons of experience on growing algae, including Carenia. And he said that, yeah, maybe we can give it a try on scale up. And then we did actually scale up. We're going to see some results soon. The next objective is into the benchtop study. So looking into, how do we absorb saxitoxin and pyrrhodinium? And we want to study three different things. We want to know how much biochar we need. So that's a dosage study. We want to know how fast that absorption is happening. That's our kinetic study. And then how much of that that sorbate actually how much it would be stored in it. So that's my equilibrium study, that the maximum amount we can absorb. So these are three things we can do. And once we know this, then we can do our column study at Mote Marine Lab. So looking into the first objective, and then this is how you would see. So we got our starter kit, 50 milliliter starter kit from FWC. Thank you, Corey Lopez, to do that and be able to do it. And we get that one as well as how to maintain it as well. So the cultures were grown from 50 milliliter to 2.8 liter flask that you would see over here to 12 liter glass carboys. And there are some struggles initially, but then we figured it out, and now it's growing very well. So now we have a growing media, and then the culture conditions are reported over here. So using the L1 media, the 12-12, the daylight cycle, and using that about 65 micromole quanta the power. Now, how do we maintain or know that the cells are healthy and happy? We would usually do it for the microscopy as well as the PAM fluorometry to find out. So far, we have been getting about 1 million or 10 million cells per liter, which is good because then we can do all of our benchtop study as well as our 80-liter tank, which would probably need 1 carboys that you would see over here, because usually in Tampa Bay, the high cell concentration would be around 500,000 cells per liter. And we would also confirm that the growth rate is similar to what published in literature, about 0.25 division per day. So now that we know that we have we can scale up and then we have a culture in hand, then we look into our biochar. So for biochar, we sourced our biochar from a commercial vendor in Indiantown, and so they can only give us the residence time 30 minutes, but the temperature, they couldn't really give us a temperature. So we had to do, to understand fundamentally what's happening, we also made our own biochar. So this is how it looked like. So this is my loblolly pine, that's the LP, pyrolyzed at 400 degrees Celsius, loblolly pine pyrolyzed at 600 degrees Celsius, loblolly pine pyrolyzed at 800 degrees Celsius. And then this is my commercial biochar that is also obtained from a pine. And everything is prepared 30-minute residence time. I thought of the question. Anyway, so and you see from and we want to know how it compares, and you see that from scanning electron microscopy, the commercial biochar, this is a different cross section than that, but you would see that the pores are getting more and more visible in 800 and 600, very close to or similar to commercial biochar. So that's my SEM. Another thing that we also did is the elemental comparison. And we see that you compare the elemental comparison of commercial biochar, and it is actually in between 600 and 800 degrees Celsius. One thing we also see, there is a little more oxygen in commercial biochar. And next, we also run the porosity, and this is very important if you are talking about adsorption or porous medium derived adsorption. So this is very important, and we see that the commercial biochar has the BET surface area 261 would be in between, very similar to 600 and 800 degrees Celsius biochar. So this gives us a very a good understanding of maybe the biochar was produced around 600 to 800 degrees Celsius. Now, then we also looked into the functionality. Again, porosity is very important for adsorption as well as functionality, because we don't know which one we actually weigh more. So we looked into the total oxygen functional group, and we see that for our lab-made biochar, the 600 and 800, although they morphologically look similar, but they are not that much of oxygen functional group compared to commercial biochar. And that is probably telling us that in commercial biochar, instead of using a complete inert environment, they might have used a little bit of partial oxidation to actually enhance that oxygen over. There's very common practice for commercial biochar processing. So that's the biochar we understand about biochar. Now let's look into how it performs. So we started our performance using the saxitoxin. We got our saxitoxin from Gold Standard, and this is the standard saxitoxin, and then we dissolve in, we get it in water, and we started with 100 ppm of saxitoxin. We did different dosage rate, different contact time, and different concentration to go through the dosage, kinetics, as well as equilibrium, and then we eventually get into the ELISA to do the saxitoxin concentration. These are some of the results that we obtained over here. So this is the dosage study. And again, the dosage study, we vary the dosage from 0 to 1 gram per liter, while we make the toxin concentration constant and 10 micrograms per liter, the contact time constant at 24 hours. And we see that the saxitoxin really loves biochar. So we get about very low dosage, like 0.04 would allow us to get about 90% removal of saxitoxin. But to get about 95% removal, we need about 0.2 to 0.4, depending on the biochar we are talking about. So that's my dosage. Now we look into the kinetics. Now it's looking into like how fast it is happening. So now we constant our dosage rate 0.4 grams per liter. We have our initial concentration constant at 50 micrograms per liter. Then we are varying the contact time to evaluate how fast the adsorption happens. And we see that the adsorption happens, if we get into the 95% adsorption, it would take about 30 minutes to 3 hours to get into adsorption. So it is relatively fast adsorption. Then the question comes to our mind is how the adsorption is happening. So we did the modeling, and in terms of modeling, there are 3 models that we tested: a pseudo first order, pseudo second order, and pseudo third order, so the Ilovich model. And the first one is basically tells us the phase absorption driven. So this is the fastest one, and then it would be chemisorption driven, and then it would also be chemisorption driven, performed by the electrostatic attraction. And we looked into that when we fit the data, just looking into the commercial biochar over here, you see that it's both pseudo first order and pseudo second order. So it's basically telling us that it is driven by chemisorption as well as phase absorption for the saxitoxin adsorption onto biochar. So now that we know how long it takes and how much dosage we need, we then went into the equilibrium study. So it's basically how much we can absorb in per gram of biochar. So in this case, we are we make our dosage constant, 0.4 grams per liter. The contact time remains constant, 24 hours. And again, it probably take about 3 hours to reach equilibrium, but in order to get everything into equilibrium, we just chose the very long residence time. So that's my 24 hours. And then we vary our initial concentration and see how when it is reached equilibrium. And these are the results for that. And once we did that equilibrium study, and you see that the QE, the equilibrium absorption capacity, the maximum absorption capacity, it varies from biochar to biochar. We can get as high as 200 micrograms per gram. We didn't go farther than 100 micrograms per gram because you would probably not see that much of a high concentration. But just like the kinetic study, we also did the isotherm model, and then there are fairly known isotherm models like Langmuir model, Freundlich model, and Langmuir-Freundlich model as well. So what Langmuir model tells us that it is a monolayer adsorption. So if I have functionality, it's a one-to-one adsorbed and then nothing adsorbed afterward. Freundlich model tell us that this could be a multi-layer adsorption. So what we also known is as a stacking mechanism. So just stack one above another. And then in many adsorption medium, if I have both functionality as well as porosity, it would actually show us the combination of mono and multi-layer adsorption. This is my Langmuir-Freundlich. And indeed, we see it from our commercial biochar. Again, I'm just saying commercial biochar over here. This basically have both Langmuir and Freundlich behavior. Our lab-made biochar has like initially was more Langmuir type, and then it reached into more Freundlich type. So that's my saxitoxin. So once we know all of these, we can come up with the adsorption mechanism, and we believe that it is happening both functionality as well as the porosity. So it is probably the pore filling chemical interaction because we have the pi-pi bonds in biochar as well as saxitoxin. We could actually experience that as well as electrostatic attraction as well. So this is all happening without cells, just pure toxin in biochar. And then we published this paper in Chemosphere early, I think the late last year, and then the issue comes out in February. So that's that. Now the question is, okay, so now it works very well for saxitoxin. Does it actually work with harmful algal bloom cells? And this is where we stumbled a little bit, because when we ran our zeta potential, we found out that the biochar, that lab-made biochar as well as the commercial biochar, those are negatively charged. And as you remember, our harmful algal bloom cells are also negatively charged, so they would not probably coagulate. So once, and if it's not coagulating, it would probably be very difficult to remove it. And then I think I don't have the data over here, but we tried only biochar, and it didn't really work as a removing harmful algal bloom cells. It does very good with the toxin, but not with the cells. Now I talked to Dr. Vince Lovko, and then we come up with that maybe we can just add a little bit of polyaluminum chloride. This is what they have been doing for other technologies, for example, modified clay, etc. So the polyaluminum chloride, it's a very common coagulant, mostly because it has a lot of Al3 plus ions, so it is a highly positively charged. So zeta potential is about plus 20 millivolt, and our thinking, what maybe we can just physically mix it. We're not combining them, so it's a physically mixing, and we tried it with 1:4 ratio for this study, and then that gives us the zeta potential positive. And then this is how the SEM look like. So this is my polyaluminum chloride, a spherical particle, and this is my biochar. They're next to each other, which is going to help you. So basically the idea becomes like, once we coagulate the biochar and making the floc, if the cells rupture and toxin will release to water, biochar will be able to capture it. So that's the current hypothesis. And then we run our coagulation study, and we have our two different coagulation or cell concentration. A high was 50,000 and the low was, high was 500,000 and low was 50,000 cells per liter. And this is how the cells look like. And these are just the triplicate concentration that you see over here. So I have the 3 control runs in the left, and then 3 what we call the mix or modified biochar on the right. So again, we did that, and then we mimic what we will be doing in the column study. So it's a 12-12, the dark and light, and then we did the cell. We did the experiment for 3 hours, 24 hours, and 48 hours, and then we did the cell count. And these are some of the results that we are finding. And you see that, so the black one is my control run, no biochar added, modified biochar. And the red one is my the added modified biochar. And then here the modified biochar was 0.2 grams per liter, which translates to about 0.04 grams per liter of biochar and 0.16 grams per liter of polyaluminum chloride. And it looked like in 3 hours, we are actually seeing about like 95%, 98% of flocculation. So the cells are removed by the biochar. Now, the question is, the cells are removed very well. Does it actually work with the toxin over here? So we did the ELISA, and we found out that for only toxin, there was a 90% removal for 0.04 grams per liter, compared to here, we are seeing about 60% removal. So there is a little bit compromise we are making. But again, maybe we can just play with the ratio and then we can increase it. Our hypothesis is maybe because now the entire thing is positively charged, not negatively charged. So electrostatic attraction is not happening. So we are actually not seeing that part of the absorption mechanism. So that would probably results us to a little bit lower absorption of saxitoxin. So that's what we have been finding so far. Now, the last part of it is I'd like to talk about is our column study. So our column study will be performed in Mote Marine Lab. So these are some of the columns you see over here. And the columns are about 1.2 meter tall and have multiple sample ports. And these columns are designed to mimic the Sarasota Bay or Tampa Bay estuary. And the tanks are housed and with a UV light, and then we can have the target cell abundance about as high as 500,000 cells per liter. And then after the column study, we are also planning on doing the cell abundance, the PAM fluorometry, and ELISA do. So that we already we were kind of planning on doing it, but then nature happens. So we have a Hurricane Helene impact. So that is, we probably remember that. So Sarasota hit pretty hard. And Mote Marine Lab also impacted. It was unavoidable. And you see some of the damage that performed. And unfortunately, that also damaged the 80-liter system over here. So, but luckily for us, we are actually relocating, or we relocated the 80-liter system from Mote Marine Lab to the Mote's Red Tide Mitigation and Technology Development Research Facility. In short form, we call the MAB facility. And this is the new system look like. And then it's more rugged and also it is much more inland than the Mote Marine Lab what we had previously. So the building is nearly complete, and we are anticipating to run those that start functioning about March or this month, and then probably start running our experiments in the next couple of months. So for that reason, I reached out to Tampa Bay, and they graciously agreed a no-cost extension for this project until spring 2026. So the project is still going. So, so I can conclude now. So the couple of things we have learned. One is we can culture pyridinium. And we can culture from 50 milliliter starters, and we are doing it up to 12 liters, and it's been doing well. We also learned that the biochar contains high porosity, it is carbon dense, and functionality. Now, depending on which temperature, it can change a little bit. And from the batch absorption testing, we see that the dosage amount, 0.2 grams per liter, required about 30 minutes to 3 hours for 95% removal of saxitoxin. And this is pure saxitoxin. It is a mixture of pseudo first order and second order reaction. And it is, in terms of the isotherm modeling, it follows more of a Langmuir-Freundlich isotherm. In terms of modified biochar, we see that modified biochar is able to flocculate and remove pyridinium cells probably pretty effectively, and the pack is doing its job over here. And we see the saxitoxin also removed, not as high as pure saxitoxin removal, but we are working on that. And finally, our column is now set up and ready to run. Hopefully, we can show you more results in upcoming months. So with that, I would like to acknowledge again Tampa Bay for funding us, and that would allow us to learn more about pyridinium, and as well as the biochar and modified biochar. And our biochar was procured from Green Carbon Solution. I'd like to acknowledge Carrie Lopez for providing us the starter pyridinium culture, as well as the recipe, and also my undergraduate and graduate students, and also the staff members at Florida Tech. And so far we have published 2 papers. There are a couple more on the way, and these are the references. And if you have any questions, we'd be happy to answer. I have my co-PIs with me, so a full team here. I'd be happy to hear your thoughts about the project and any questions that you might have. Thank you.
Thank you very much. All right, do we have any questions for the project team? If you'd rather ask verbally, just unmute and we'll try to manage that as well. I see a hand
I have a
up.
question. Hi, Ed.
Fee, how you doing? Great
Good.
presentation.
Thank you.
Just thinking more of like longer term and how the maturation of the project might proceed. I know it might be beyond the scope of what you're thinking about, but my question is just kind of tailored to your entire team to think about in applying either biochar or modified biochar, and seeing potential effects on both saxitoxin and settling of pyridinium cells. Have you thought about maybe looking at a mixed algal culture in the future to see, after the effects of the biochar and the settling occurs with the pyridinium, that available carbon and nitrogen, what impacts it might have on availability to other algal species that might be in the system, and how that could affect the dynamics of blooms moving forward? So just throwing that out there, food for thought. And if you have any comments on it, I'd appreciate it.
Absolutely, absolutely. So I'll start. Me and Vince, we talk about this topic more frequently. So yes, this project will end with 80-liter column study. The next logical study is our mesocosm study that will be performed at Mote Marine Lab. Now, currently, I mean, this project will end, but we have recently awarded an EPA project where we will be able to do mesocosm study, but for Carenia, and maybe if we have funding, we can do a mixed culture of Carenia as well as pyridinium to see how it works. And then eventually, I mean, we have another project under review that is with FDP, and that is more of a field demonstration, but that is for more of a freshwater, because we really wanted to do seawater, but they brought us back to like, okay, no, we have to do freshwater because that's where the funding is allocated from. So we are thinking about it and see where it goes. I mean, this is just the starting. I'm glad that we are still heading in the right direction. The hypothesis, we have to work around the hypothesis a little bit, and this is very common for low TRL technology, but it is moving forward. Vince, you want to add something else?
Yeah, I'll just say that it is outside the scope of this current project, but certainly we have various projects that are using biochar for HAB remediation, and certainly, as Tawfiq mentioned, some future analyses will be what is the impact on other phytoplankton species within the water column. Now you're talking about that long-term, deposition of the carbon and the whatever is sticking to the carbon in the bottom. Tawfiq could probably answer this better than I can, but I think I understand there's a pretty long half-life, at least as far as we know in terrestrial systems, at least of biochar. I don't think we know what it is in the aquatic system.
And that's one of the tasks for our EPA project. We're going to find out in a couple of years. I know the half-life of biochar in soil is about 1,000 years, but half-life on water could be a little different, and it would differ from seawater to fresh water. So this is one of the tasks we are going to do for our upcoming EPA project.
Yeah, no, I'd also emphasize that the amount of biochar that we're using, much like the amount of material that we're using in some of the other flocculation and sedimentation technologies is quite small.
There was a follow-on to that about the impact of various diatoms or pseudonychia rather than K. brevis. So how does I think this is kind of a follow-on to Ed's question from Maya. Is it the interaction with other algal species in the ecosystem?
I can start, and then I'll probably hand it over to Vince. He has more experience with different species, but recently we were awarded the US HAB CTI project, and this is interesting because now we are able to test our single biochar material for 3 different species, so Karenia brevis, Pyridinium, as well as Alexandrium, and we are finding that it would be very good to have one solution for all of them, which is true, but the dosage is different for each of them. And as well as how much they, some of them actually, the kinetics is much faster than the other ones. And it makes sense because if you look into, brevetoxin, PBTX3, a very long molecule versus your saxitoxin and a small molecule. So you have a small molecule versus long molecule, we would actually have different the kinetics we are talking about. So it is understandable, and we are working on that. So one thing that we are not doing in that project is to mix those cultures. So that this is something we didn't propose in that project, but yeah, it could be interesting. Vince, you want to add something else?
Yeah. Well, as far as that goes, I mean, and this ties back to the previous question a little bit, is in the future in some of these other projects, we do intend to use mixed assemblages, if possible, natural, blooms, both in the Microcystis project as well as the Karenia project. As far as the one question from Maggie in the chat, as far as removed, what we mean is it's absorbed to the particles and it's sunk down to the bottom. There is no intention of then filtering it to remove the cells. The idea, much like the clay flocculation, is that the cells are bound, they go to the bottom and they can't come up to the surface to photosynthesize, so they die. As far as the other question from Maya, I believe, so is that in terms of like the effectiveness on pseudonychia, like using it as a mitigation for pseudonychia? If so, we have not tested diatoms. I don't think we've really thought about diatoms, but but it could
Well,
be a future project.
I think
could
you
be.
kind of raised the issue. Like that's what we see basically is these mixed assemblage blooms pop up, and we're sort of interested in the trophic cascades that that we kind of saw in Indian River Lagoon, that
Mhm.
kind of went to Aroambria and some of these other species, and we're seeing signs of that in Old Tampa Bay. So
Mm.
I think Ed's question was kind of wondering like what kind of nutrient availability or like other kinds of follow-ups, like how this might affect the dynamic feedbacks in the system for some of these other species that we're also observing in Old Tampa Bay.
I sense a very let's see. I'm a
Future
researcher.
project.
I sense a research question. So no, Maya, we haven't looked into it. Me and Vince, I think based on my experience with last couple of years about biochar and Vince's experience with many years with harmful algal blooms, we can put together a hypothesis. But we cannot probably give you an answer without testing it.
I think we're more trying to seed you in the direction of some of like the less attention grabbing algae blooms that we're all kind of dealing with. So not everything is Karenia or, you know.
But that's where the money is.
That's what the money is, unfortunately, yes.
And
But we're telling you where the needs are too.
we'll go with the last question before we move on to the next presentation, but Maggie had a follow-up question. Would the pyro cells just insist if you sink them and then reemerge next year?
That's a very good question, and something that we deal with Karenia and some of the clay work and other work we've been doing as well is, can they eventually escape? And so where we've gone with some of the clay work is we've started adding in algicide so that we actually we can kill the cells. We have talked about that, and in some little intern projects we've worked with that a little bit with the biochar as well. So what we could see in the future is like more of a combination, like using biochar to adsorb toxins, maybe using something like clay if necessary to help sink it, and possibly reduce the amount of biochar we might need, and also using an algicide to make sure we're killing the cells. So you bind the cells, you kill the cells, you adsorb the toxins, you take it all to the bottom. So yes, that is potentially a concern and something that we need to look into once we determine how well this works.
All right, well, thank you very much. Great discussion, great questions. Look forward to the next steps in your research.
All right. Well, thank you very much.
All right. So we're going to move on to identifying Tampa Bay habitat protection and restoration opportunities using CAP land cover. Casey Craig with FWRI. All right, we're good.
Okay. Yes, so my name is Casey Craig. I am an assistant research scientist at the Florida Fish and Wildlife Research Institute, specifically within the Coastal Wetlands Research Group. And today I'm going to be presenting on a project that we recently did with TBEP that is GIS based, where we're really looking for habitat opportunities within Tampa Bay. And so today my presentation is titled Identifying Tampa Bay Habitat Protection and Restoration Opportunities Using CAP Land Cover. All right, so to begin, we're actually going to be talking about the Habitat Master Plan update, which I know everyone here is very familiar with. It was published in 2020, and specifically what we're going to be talking about is Section 5 of the Habitat Master Plan update, which outlines an opportunity assessment workflow of GIS analyses using land cover data with the intent of identifying habitat protection and restoration opportunities within the Tampa Bay watershed. So the analysis produces a map that looks like what you see here, and so it is color-coded based on the opportunities. And so this analysis that was included in the Habitat Master Plan update was originally based off of the Southwest Florida Water Management District's land use and land cover layer for 2017. More recently, however, a higher resolution land cover layer was published by NOAA. So NOAA's Coastal Change Analysis Program produces standardized high resolution land cover data sets for the coasts of the US and its territories. And so these are really suited for regional use. But more recently, a even higher resolution, 1 meter resolution CCAP data set for Tampa Bay was created. And so this is sort of, Tampa Bay was one of the sort of, I guess you could say, pilot areas where there's, they started with this 1 meter resolution data set. And so this was using imagery that was captured in 2021. So compared to the district's land use and land cover data set, which I'll be referring to as LULC, just for ease moving forward, versus CCAP, there are a lot of differences. And so CCAP will be talking about the NOAA land cover layer. When we say LULC, we'll be talking about the district land cover layer. So when we compare those, we do see that CCAP is a much larger data set. It's almost 4 times the size of the LULC data set. And that's largely because it is such high resolution. We're talking 1 meter for a minimum mapping unit versus 0.5 to 5 acres for the minimum mapping unit in LULC. And so that variation in the LULC is due to the habitat type. So different habitat types are mapped with minimum mapping units. Conversely, however, it is mapped less frequently, with data sets expected every 10 years compared to a frequency of about 1 to 5 years for the LULC. And it uses a different classification system than that of the LULC, which uses FLUX, the Florida Land Use and Cover Classification System. But the CCAP data set has a fewer number of land cover classes. So you can see that there are kind of pros and cons to both. So we use this high resolution CCAP data set that has been published to redo the GIS analysis that's described in Section 5 of the Habitat Master Plan update, with the goal of identifying habitat opportunities that might have been missed in the previous analysis. So these are especially in areas that may have been classified as development before. So to do this, we needed to leverage the high resolution capacity of CCAP, but also the abundance of land cover classes that's offered by LULC. So we crosswalked those data sets. So the diagram that you see here is a very simplified representation of how we did that. But if you're interested in the finer details, unintended, they are described in the report that I just dropped in the chat. Essentially, what we did was we created what I call a mega crosswalk that translated each CCAP land cover class to a unique LULC land cover class. And so then those could be binned into Habitat Master Plan Update land cover categories. And so those are what you see on the bottom here. That's native habitat, restorable habitat, development, and subtitle. The analysis focused on native and restorable habitat categories, and that is because existing development was removed really because the majority of these lands are under private ownership, or they are being used for public infrastructure, which makes them unsuitable or impractical for cost-effective habitat restoration. Subtitle habitat was also left out because most of the bay bottom is state-owned and therefore under a level of protection as sovereign submerged lands, with some of these areas offering additional protections of an aquatic reserve or wildlife refuge designation. So we can take those, we took those layers of native and restorable habitat of those, binned land cover data sets, and we're able to analyze them through a 3-step GIS process. And so the first step identified existing habitat opportunities. The second step identified the proposed habitat opportunities. And the third step identified habitat reservation opportunities. And so we're going to get into kind of what each of these means and how they were identified for the rest of it. So again, taking those data layers of native and restorable habitat, I intersected them with an existing conservation lands layer of Tampa Bay. So this existing conservation lands GIS layer includes areas such as state parks or preserves that have an existing level of protection through conservation easements or public ownership. So leveraging the predictability, protection, and control of the areas really makes them ideal and cost-effective targets for conservation, restoration, and management. So this intersection process identifies native habitats that are good candidates for management and enhancement activities. This could be things such as nuisance species control or minimizing anthropogenic impact, as well as our existing restoration opportunities. And so both of those are illustrated in the map. The lighter green is native opportunities and the darker green is restorable opportunities. When we look at the acreage breakdown, so in these tables that I'll be showing you, this is the actual CCAP land cover description and relative acreage extent that was identified during this part of the analysis. And so what I want you to get from this table here is that the management and enhancement opportunities of our native habitats reside primarily in our palustrine forested wetland. So this was about 50% of the opportunities identified, with mixed forest coming in at the second most covered land cover category, which was at 19%. So if we dive a little bit further into those existing restoration opportunities, we can take that analysis one step further by trying to identify which type of habitat restoration would be most suitable for a given area, given its one, soil suitability, and second or two, its position within the coastal stratum. So the coastal stratum here is defined as lands that are within 5 feet of mean lowest low water. And so when we do that workflow, which you see in the diagram, the result is the map on the right-hand side. And so when we look at this acreage breakdown, you can see that freshwater wetlands provide the largest opportunity for restoration, with pasture lands contributing the largest majority of those lands. The most expansive land cover class with existing restoration opportunity in tidal wetlands, coastal uplands, and non-coastal uplands was mixed forest. As I mentioned before, our native habitat opportunities. And so mixed forest is really defined by CCAP as trees, areas where trees are taller than 5 meters, and vegetative cover is a little bit over 20%. So something else that you'll notice is that our existing restoration opportunities are more densely concentrated on the eastern side of Tampa Bay. And that is really reflective of just sort of where the development is. Obviously, we all know that the Pinellas County side of the bay is incredibly developed relative to the other parts of the Bay watershed. So if we look at some examples that were identified using this part of the analysis, we have 2 here. In the left image, we're looking at Felipe Park in Safety Harbor. And that gray overset or those gray polygons are actually the opportunities that were identified using the 2017 LULC layer. So this is really highlighting some areas that were, again, outside of the previously identified opportunities that we found here. So we have Felipe Park again on the left side, and on the right side, we have some medians and vegetative coastline along the Courtney Campbell Causeway. And so the light green patch that you see here is the native opportunity, and the dark green patch is restorable opportunity. Looking at another example, we have this enhancement and restoration opportunity, which was identified in Meadowlark Park in Plant City. So this is a little bit further west into the Bay watershed. Again, the gray overset is the previously identified opportunities. And in this image, the dark green is actually a lake that was not an opportunity that was identified through this process. And lastly, another example just to show some of the existing opportunities is this enhancement and restoration opportunity at Fort DeSoto Park in St. Pete. And something that I really want to highlight here is you can see that there's a parking lot in the middle of this green patched area. And the CCAP data set is so fine resolution that it's actually able to pick up these small little gaps between parking lanes as habitat that can be eligible for enhancement or restoration. So now that we've talked about step 1 of the analysis, let's dive into step 2, which involved looking at proposed opportunities. So TBEP and subject matter experts created a proposed conservation lands layer in the Habitat Master Plan update, which represents the best judgment of priority parcels of land that should be targeted for acquisition. So by intersecting this on native and restorable habitats, we can determine suitable locations for proposed conservation of native habitats and proposed restoration opportunities within Tampa Bay. And so these proposed opportunities are illustrated in the map that you see here. Again, the darker color represents restorable habitat, while the lighter color represents native habitat. When we look at the acreage breakdown, we find that of the proposed conservation opportunities, 53% of these lands were classified as palustrine forested wetlands, which is similar to what we were seeing with the existing native opportunities. With another 20% being comprised of mixed forest land cover. Of the proposed restoration opportunities, there was a little bit more of a split, with cultivated crops comprising about 32% of the opportunities identified and pasture lands comprising 30%. And again, you're going to notice that a lot of these proposed opportunities are concentrated on the western side of the bay. When we look at a few examples of what were identified through this analysis, one example is this area, which was classified as palustrine emergent wetland in 2021 when imagery was captured. But as you can kind of see now, in 2024, it's actually the Esplanade at Azaria Lakewood Ranch Golf Course. So this is a parcel that was obviously not be practical to conserve or restore due to its use for hard development at this time. And so this really just kind of highlights the dynamic nature of land use change within the Tampa Bay area, especially within the last 5 years. Because between this 3-year gap of when imagery was acquired versus when it was published, or the data set itself was published, we already have this common trend in land use change. And so another example of that is the single largest proposed restoration opportunity that was identified through this part of the analysis. And so you see that here in that hatched blue, this was classified as croplands in 2021, but this is now the Duke Duet Energy Solar Farm. So again, this would be a parcel that's no longer practical to conserve or restore due to its use for development at this time. An example that's a little bit more practical is this restoration opportunity that was identified along Meadow Point Boulevard in Western Chapel. Again, what we're seeing with what we saw with the Port Soto example and some of the other opportunities is that this is in the road medians. So again, just characteristic of the high resolution. Let's see what they say. So we talked about steps 1 and 2. Now we're going to be diving into step 3, which involved identifying preservation opportunities. Lands within the coastal stratum, as a reminder, are defined as lands that are within 5 feet of mean lowest low water, are already experiencing impacts from sea level rise, which are expected to increase in magnitude by 2070. So reservation lands are unprotected lands within the coastal stratum that represent coastal areas in greatest need of conservation action through public acquisition or easements. And so the proposed native and reservation opportunities are illustrated in the map that you see here. And when we look at the acreage breakdown, we see that there are over 19,000 acres of reservation lands identified, of which most of this opportunity is concentrated in native habitats, which is about 72%, and the remaining 28% of opportunities were in restorable habitats. Within those native habitat classes, the largest reservation opportunity was common trend, palustrine forested wetlands, offering 32% of the opportunity, with estuarine forested wetlands falling behind with 30%. When we look at the restorable habitat classes with the largest reservation opportunities, we find that developed open space really dominates this category, comprising 45% of those opportunities, followed by mixed forest at 20%. And so this is characteristic of sort of how development is more densely concentrated around our coasts. That's unfortunately also where people are also at risk for sea level rise impacts. So if we look at a few examples that were pulled out from this part of the analysis, first we have this opportunity that was identified on Treasure Island Beach. This is just south of the Johns Pass Bridge. If you're familiar with the area. And so you'll see the beach section and then a lot of smaller parcels on the intercoastal side. And so a lot of these are actually homeowners' lawns. So here's another example of what was a very common find for what a lot of our habitat opportunities look like. So this is in a neighborhood, or this is a neighborhood near Eagle Lake Park in Largo. And so these are all private homeowners' lawns. But again, because we had this fine resolution data set, we were able to pick up these areas as opportunities for reservation or restoration. One more opportunity that was identified is this native and restorable reservation opportunity along the Alafaya River. So how did the CCAP data set do relative to the previous analysis that was completed using the LULC data set? So in this table, we're looking at a comparison of the two data sets. So in the top, you have your LULC values, and in the bottom, we have the values for the 2021 CCAP-based analysis. And so the first table column is a little bit different this time. Rather than looking at the CCAP land cover class, we're looking at these habitat groupings, which are coastal uplands, non-coastal uplands, forested freshwater wetlands, and non-forested freshwater wetlands, with their respective acreage extent of each in the remaining columns. So when we look at this breakdown, it's pretty clear that CCAP was able to identify more of the habitats than the 2017 LULC-based analysis was. This is evident by, if you look at the three columns on the left half of the table, you'll see that each of those CCAP columns identified more habitat than the previous analysis. However, one notable difference is when you look at the right half of the table, which is our restorable opportunities, you'll notice that there were fewer restoration opportunities identified using the CCAP data set. And so this could be for a multitude of reasons. This could be because of habitat expansion or contraction. This could be to land use change, but really without diving into each parcel individually, you can't really know what the driving factor was for that. So if we, again, are doing a comparison of the 2017 analysis versus the 2021 analysis using CCAP, we found that over 11,000 acres of newly identified habitat opportunities were found. The native land cover class with the largest opportunity across the board was Kallestrom forested wetland. That's not really a surprise because when we went through each of those stepwise, that was a common trend. Things were a little bit different, however, for our restorable habitat types, with developed open space dominating the proposed and reservation opportunities. While mixed forest dominated the existing restoration opportunities. So to review, the analysis using the CCAP data set proved beneficial for identifying habitats in the developed spaces. A little more than 11,000 acres of habitat opportunities were newly identified using the CCAP data set. These were concentrated in urban and coastal areas where development is also abundant, suggesting that the CCAP analysis was successful at identifying areas previously considered development. Over 99% of these new opportunities were smaller than 1 acre in size, kind of like what we saw with those areas identifying people's lawns. So again, this could be a result of habitat expansion, for instance, natural forest growth. This could be due to differences in delineation methods because they use different minimum mapping units, or it could be a change in land cover or land use. For instance, croplands that were sold for hard development, like we saw with the solar farm that was identified. Many areas that were identified as candidates for enhancement or restoration are already in use for development. like I mentioned, that golf course and solar farm, which really highlights the dynamic nature of land cover and use change within the Tampa watershed, particularly within recent years. I'd say probably the last 4 or 5 years or so, especially. And a common trend was for opportunities to not fall on land directly being used for development at this time, but maybe have future plans for development. This could be things like unoccupied land adjacent to the solar panels, or sort of a buffer area around like a retention pond. So with that, the final analysis produces a map that looks very similar to the one we saw on the first slide, but you can see this updated map here. And with that, I can go ahead and take any questions.
Thank you. All right. We have one comment in there, just Maya just said, just because something is a golf course now doesn't mean it isn't a good restoration candidate in the future.
Exactly.
Agreed. Agreed.
Definitely.
Do we have anybody who wants to just unmute and ask, or?
Yeah, I can ask a question or make a couple of comments as well. This is Randy Runnels, Tampa Bay Aquatic Preserves. Thanks for looking into this. And I couldn't tell, I was straining my eyes to see there's a 80-acre parcel north of floodplain on Lake Tarpon, north of Chestnut Park. Was that in one of the areas targeted for conservation? Because we got a lease on that about 10 years ago to manage that for conservation. And I didn't know if that was up to date or not. I couldn't really tell if that was, it's kind of a wedge shape just north of Chestnut Park on Lake Tarpon. And I was wondering if you know the status of that in your database.
Existing conservation land data layer that I pulled. I did, Marcus let me know that it's probably a little bit outdated from when I did the analysis last year. So I would bet that that is not included in this part of the analysis, if it's even more recent than that.
Okay, yeah, because we do, I mean, even though it's state land and we're state agency with DEP Tampa Bay Aquatic Preserves, we have to get a lease to be the official managers. But we realized when the state realized they owned it, it had been donated decades ago, and we didn't know the state owned it until people asked about it. When we realized that the state owned it, we asked to get a lease, and so we're managing that. And I also want to comment on the acreages. I'm sure this is kind of preaching to the choir here, but some of the small acreages in Pinellas County, even though the acreage is not great, some these small habitat patches in an urban landscape can be really important. And that 80 acres, we see birds foraging that probably nest on the saltwater islands in Clearwater Harbor, foraging over there. They have a short 6-mile flight to low salinity foraging areas and things. So we're trying to look at that urban landscape. And I think this is a good start on identifying other parcels, but I hope you'll continue to engage with on-the-ground resource managers about some of the particulars of these parcels, because on some of the dredge material islands, we're converting invasive plants to high marsh and things like that. The migratory neotropical songbirds really make use of those when they're trying to navigate through an urban landscape to rest and feed and all that. So, and the other comment I want to make, because I've seen this in the past, I've seen people whose bread and butter is restoration, prioritize encouraging acquisition of restorable habitat over acquisition of intact habitat, because of the restoration potential. And most of the restoration, I mean, it does add ecological function, but it rarely has the ecological function of intact, undisturbed habitat. So I just hope if you see people falling into that pattern of trying to say, well, this is more restorable than that because that's intact. Well, it's a good idea to keep in mind that the intact habitat could be impacted and needs to be preserved as a high priority. That's it.
Thank you. Any additional questions, comments?
We've got Mark with his hand up.
Okay. Mark?
Thank you, Kelly. Can you hear me okay?
Yep.
Okay, good. Thank you. So Mark Schrammick, NOAA National Marine Fisheries Service. Appreciate Randy's comments as always. I'm just kind of curious though, Casey. So those individual small privately owned parcels, one thing in my experience working in the federal permitting arena is that, you have these subparcels of land, whether it's a mangrove, wetland, or tidal marsh. And I find that when they're on, individually owned platted parcels, they're often proposed for impact. So I would use caution and maybe potentially identifying those as candidate habitat restoration sites. I do agree with the ecological value that Randy pointed out, but in my experience, it seems like, if Peter has a seawall and then Jessica wants a seawall adjacent to that, usually it's at the expense of the wetland habitat. So just my opinion, but I appreciate the comment and glad to see this data is being utilized. I think that's great. Thank you.
I think that's a really good point, Mark. Not sure why it's echoing now. But I also would just say, like, there's different tactics that we can employ. I'm, pretty convinced by the Douglas Tallamy argument that we have to have sort of outreach and restoration campaigns that are focused on things like backyard habitats. And so it's a different approach to restoration, and it certainly has different vulnerabilities, as you're rightly pointing out.
Yeah, living shoreline. We're always a big advocate for living shorelines as well. So, but again, getting the buy-in from the individual homeowners, often it's a challenge. But when you have coastal resiliency demonstration projects, I think they're more effective. Thanks for the thanks for the chance to weigh in. Great project.
All right. Any additional questions?
Yeah, I just wanted to chime in too. First of all, I wanted to thank Casey for doing this analysis because we gave it back to her several times, asking her to do revisions on things like the lookup table and how we categorize different things to do that crosswalk. So this was no small part or no small effort on her part. So definitely appreciate that. But I guess I just wanted to also kind of step back and contextualize this a little bit more. So as Casey mentioned, this is really stemming from the update to our Habitat Master Plan that was completed in 2020. And so I know this group probably saw many presentations on that ad nauseum, basically. So hopefully, we're kind of familiar with what that is, but just to remind everybody, we essentially have as a program short-term targets, so 2030 targets and long-term goals out to 2050 of coverages and native habitat, acreages for native habitat that we want to achieve as a program, where we're trying to sort of seize the existing or remaining opportunities where there's either native habitats that haven't been acquired, or there's areas that we know that could be acquired for restoration. And so we have from that Habitat Master Plan those areas identified, and I think everyone sort of got the message of this talk where we have now this higher resolution data set that we can use to really identify these additional areas that may not have been covered by the more coarse resolution land cover data that was used for the original Habitat Master Plan update. So we have actually that data set that is available from this analysis, this updated analysis. We have it. We haven't made it available, not for a particular reason. We just haven't, I think, put it on our website. So if anybody wants that to sort of do the deeper dive into this data, we can certainly make that available. Reach out to myself or Casey. We'd be glad to provide that for you. But again, this is a first step towards a more intentional restoration effort. This is another tool in the toolbox. It's a sort of a high level assessment of like, this is where we're at on the landscape. Obviously, if you're choosing a place for restoration, go to the site, learn more about it. But this is like your first cut. But we do hope that there are going to be some additional opportunities exposed from this data that weren't made accessible in the first round of the Habitat Master Plan update. And one last thing I'll say about it is I've heard recently there is going to be a release of the LULC land use land cover data from the Water Management District, an update to that very soon. We haven't had an update to that since 2020, and I think the data we have online for the Habitat Master Plan update for these opportunity areas are based on that 2020 layer. So do expect an update to that pretty soon, hopefully in the next couple of months, to sort of see where we're at in terms of our targets and goals. So that's all I wanted to say about that.
All right. Well, thank you. And we'll go on to the next presentation. Thank you, Casey. All right, nutrient fingerprinting using chemical tracers to identify sources in the Tampa Bay watershed. A results update. Cassidy Troxel with FIT.
Oh, it's FIU.
FIU.
Yeah, it's okay. It's okay. Yeah.
Marcus.
That's my fault. Sorry.
That's okay. It's okay.
That's
Totally
okay.
fine. But yeah, thank you. So hi, everyone. My name is Cassidy Troxel, and I'm a research assistant professor in the Institute of Environment at Florida International University. Exactly almost a year ago, I presented some of my dissertation work and presented pretty much the proposal of this project. So I'm excited to show what myself and Ocean Conservancy has been doing over the last year. So I'll get started. We have a lot to get through. Okay, you can see the screen good? Is it?
Not yet.
Oh, you can't see the screen? Okay, there. You can see the title slide now?
Yep.
Okay, perfect. Thank you. Okay, so as we all are aware, nutrient pollution is a major environmental concern caused by the excess influx of nitrogen and phosphorus into the aquatic ecosystems. They often come from urban, agricultural, and industrial sources and lead to eutrophication, triggering algal blooms, oxygen depletion, and a variety of cascading effects. Managing nutrient inputs, especially distinguishing between point and non-point sources, is essential for preserving water quality and maintaining the health of aquatic and marine ecosystems, especially in coastal environments where we have increased urbanization and sea level rise that can exacerbate these pollution sources. So the Tampa Bay watershed as a whole, which includes the Hillsborough Bay, which is the study area of our focus of this project, is influenced by nitrogen inputs. Nitrogen acts as the limiting nutrient for aquatic productivity. The Tampa Bay Estuary Program does a fantastic job compiling all these different source contributions for different bay segments. So when picking out an area to do this study, they made it really easy. We decided to choose the Hillsborough Bay segment in the Hillsborough River. This bay segment has the highest nitrogen allocation of any bay segment and has a variety of different nitrogen sources throughout it. And one of those significant ones is the non-point sources. So that's really why we chose this area and why we're employing the technique into this area. So really what we wanted to do with this Tampa Bay nitrogen fingerprinting project was to specify the contributions of point and non-point sources in the Tampa Bay watershed, specifically the Hillsborough River, where it from the most northern part to where it discharges into the bay. So pretty much we broke this project into four main tasks. The first task was to establish baselines. So using historical data and monthly collections, we were going to characterize the wet and dry season to get baselines of the different nitrogen species in the Hillsborough River. And then we decided on 3 techniques to really hone in on the different sources. So we wanted to do the nutrient analysis, again, to get all those different species. We chose chemical tracer analysis, which is using organic chemicals that are specific to point and non-point sources of pollution and quantify those throughout the system. And we also chose to do some isotopic analysis where we use the N15, N14 ratio to provide another layer of data and further conform the point and non-point source pollution that we see. So after doing this baseline, which task 1 and 2 is what I'm going to share with today. Task 3 and 4 fall into our phase 2 of the project. After this phase 1, we're going to hone in on the hot spots we identified throughout the Hillsborough River, collect directly from point and non-point sources, get the concentration of the chemical tracers, nutrients, the isotopic ratios at those, discharge points, whether it's collecting from septics, whether it's collecting groundwater near them, stormwater runoff from different areas, collecting samples from wastewater treatment plant discharge, reclaimed water, getting those concentrations, collecting flow, and doing some specific source apportionment in this river. So we chose 23 locations throughout the Hillsborough River, up from the most northern site in the Green Swamp area. Honestly, the site that we could most easily get to, way up there, out to its discharge point into the Hillsborough Bay. There's a variety of different land uses. It becomes more rural up in this area with a few ag, reclaimed water usage sites, wastewater treatment plants, all the way down through the more residential area into the urban city environment. So from May 2023 until January 2025, we've collected 20 sampling trips, which has totaled over 460 samples, where we've done all the basic water quality parameters on the nutrients and the chemical tracers. And we've identified some real hotspots, and which are interesting throughout the study. So let's just jump into some data. Of course, we did basic water quality parameters because we wanted to get the baselines for the wet and dry season. When we're collecting, we always see if this is like typical, wet, dry season conditions, helps us when we further interpret our data. So salinity, we see a gradient as we go from each of these graphs from left to right. Left is up towards the Green Swamp area. The right is down where it discharges. So each of these graphs are set up this way so you can see the trends. Of course, we have some saltwater intrusion into the river, less in the wet season as we have more fresh water coming into the river and being pushed out to the Hillsborough Bay from rain. But yep. So dissolved oxygen, which this is typical of wet versus dry season, we have elevated temperatures, lower DO stormwater coming into the system. So we have significantly lower dissolved oxygen in the river throughout the wet season. And then we have FDOM. So this is important. FDOM is a proxy for organic matter. This is fluorescent dissolved organic matter. So higher concentration of FDOM, more organic matter that you have in the system. If anyone's ever seen this river, it does have like a brownish hue to the water, especially up in these more rural areas. So this is going to be important when I talk about nitrogen. Because we do have a lot of organic matter in this system, and we also have elevated concentrations of it in the wet season. Okay, so the nutrient analysis we conducted was total nitrogen, nitrate, ammonia. Then we subtracted the inorganic nitrogen species from the TN to get the organic nitrogen. We did total phosphorus, soluble reactive phosphorus, total organic carbon, dissolved organic carbon. I'm going to primarily focus on the nitrogen species because that's why we're doing this project. But okay, as we can see in the wet season, we have higher concentrations of TN throughout the entire system. We have a trend from the upriver down to the river decreasing TN. All of the sites except for the first location were below the nutrient numeric criteria for the Hillsborough Bay. But by the time it gets to the Hillsborough Bay, they are well below those concentrations. So what I want you to note here is that the organic nitrogen, which again, we took those inorganic nitrogen species, subtracted from the TN to get organic nitrogen. Organic nitrogen, it refers to the nitrogen atom in an organic compound, which occurs as part of organic matter in soils, plant materials, animal matter, and others. But this organic nitrogen does make up a significant portion of the total nitrogen. We do see elevated concentrations in the wet season compared to the dry, but it doesn't tell the whole story. So we do see higher concentrations of inorganic nitrogen throughout different areas of the waterway. So again, inorganic nitrogen is just nitrogen atoms in inorganic compounds, which includes the ammonia, nitrates, and nitrites. Inorganic is immediately bioavailable, while organic nitrogen requires some mineralization before it can be used by the aquatic ecosystem. Of the inorganic nitrogen species, ammonia is the most bioavailable, as it does not require any reduction before assimilation into the aquatic ecosystem, whereas nitrate and nitrite, they do have to be reduced before plants and algae can take them up. So we're focusing on the inorganic right now. We do see nitrate higher up at the top of the waterway. Decreases, elevates slightly as you go through the urban environment, but it is highest up at the top of the river. We see some nitrate. We do have a significant difference between the wet and dry season. Again, that very top spot at the top of the river there. And as you go through the urban environment, we do see elevations. Ammonia and ammonium, we do see the same trend that we see in nitrite. We have higher concentrations up at the top and lower as you go down, but spikes back up when you go through the urban environment. And we do see elevated concentrations in the wet season. Oh, and just a little brief blip of the total phosphorus and SRP because we did it. So we do have higher, significantly higher concentrations of total phosphorus in the wet season compared to the dry season, as well as the soluble reactive phosphorus. Okay, now on to the chemical tracer analysis to kind of untangle some of what we're seeing. So I did create a chemical tracer method where we have different stormwater, agriculture, wastewater, potential septic tank tracers. These are all chemicals that I found in literature, have previously detected and added to one single method where you take 10 mils of a sample. 10 mils of a sample, it gets trapped on this column, spits into a triple quadrupole where it is detected, and we get some really nice detection limits. This method does only take 15 minutes, so filter your sample, put it on to run, and you get information about a variety of sources in a quick and short time. So yeah, it's super nice, and I look to expand on this method further, but for now, I highlighted the compounds that we have higher than 50% detection rate among our samples for the wet and dry season. I've highlighted a couple in specific. The light blue are potential stormwater tracers, and I'll go into the rest as we go. So jumping right into the stormwater tracer 6-PPD, which is a tire additive. People are interested in this compound for other reasons. I'm interested in it as a stormwater tracer, but it is notable to mention that this is the first detection in like the Southeast for this compound. That is a hot topic right now because it's toxicity to coho salmon and their salmonoids. So we will be publishing a paper at the end of this month on 6-PPD in the Hillsborough River in the Tampa Bay watershed, which I'm excited about. But we do see elevated concentrations as we go through the urban environment in the wet season specifically. We see similar trends with DEET and prometone. DEET is a pesticide that's sprayed to control mosquitoes, and prometone is a wide coverage herbicide that's not used in ag, but more for like weeds and stuff along roadways as well. So we did find these as pretty good tracers, and their concentrations do match up with the rainfall patterns. For agriculture tracers, we decided to choose atrazine and atrazine desisopropyl. We do see higher concentrations of atrazine during the dry season, which is typically when crops are being sprayed for different types of things. But interesting enough, we do see atrazine desisopropyl, which is its degradation product. We do see that at a higher concentration in the wet season, which is my hypothesis that, this atrazine that's there, it is getting degraded from the environment, from the sun, and as it gets washed into the river, we do see elevated concentrations of it in the wet season. I do want to add a couple more. Atrazine is being phased out from use, but Metachlor and its degradation products are next on my list to add to this method for additional agriculture tracers. And then into the wastewater tracers, sucralose and carbamazepine are really good tracers for legacy, reclaimed, and some fresh wastewater, but sucralose does not get removed. It does not get degraded from many of the treatment processes, and so it stays around forever, to where carbamazepine, it does have a half-life of 3.5 years. In Miami, we've set thresholds actually for concentrations of sucralose. If sucralose is above 120, we start to see some water quality degradation. If it's above 500, we do have impacted waters. It is my goal for every single one of these tracers, whether it's the stormwater, wastewater, ag, I want to create these thresholds for this river so that way, you know, we have baselines to go off in the future. But yeah, so we do see concentrations of the sucralose and carbamazepine elevated at the top of the waterway and down at the bottom. Again, there's some reclaimed water usage up here, as well as some wastewater treatment plants that discharge, as well as down here, there's another wastewater treatment plant that discharges into the Hillsborough Bay. So I'm not surprised seeing this, but these are pretty high concentrations. The Miami thresholds are 500 nanograms per liter. We have detections up to 22,000 ppt in the Hillsborough River. So the thresholds will definitely be different. For caffeine, we do see significant difference in the wet and dry season, and we do see elevated concentrations going throughout the urban environment. Caffeine is a good tracer for new, fresh wastewater. It does have a half-life of 5.3 to 24 hours. So once it's in the environment, it will degrade. So for detecting it, we do have new, whether it's septic influence or raw sewage, we do have some influence on the river. There's not too many septic tanks in this study area, so I would not attribute it to that. And other tracers. So I initially suggested these as septic tracers, this the triphenyl phosphate family. But I'm more thinking it's a stormwater tracer, because we do see elevated concentrations in the wet season down towards the bottom of the waterway, like we see some of the other stormwater tracers. Not ruling it out, but these compounds, this class of compounds, need to be further understood as well. But Irrigrol is just a compound added to boat antifoulant. So we do see elevated concentrations of Irrigrol down towards the Hillsborough Bay, which makes sense. We have more boating activity. Actually, I wouldn't say that, but we do have a lot of boating activity at many times of the year, but we do tend to have more tourists in the summer months. But yeah, so these are tracers, not necessarily of nitrogen sources, but we'll see. So we did some correlation analysis, and we do see some correlations between the chemical tracers and the different nitrogen species. It does show some weaker correlations, which may indicate more localized contamination sources rather than widespread transport. I don't want to get too much into these because I want to stay on time, but happy to talk about these further. In the wet season, we do see stronger and more widespread correlations between the organic contaminants and the nitrogen species, likely due to the higher runoff, increased stormwater contributions, and more wastewater influence. Yeah, continue rolling because I'm not sure what time I started, but I want to stay on time. So I threw these into a multivariate analysis. So I did a principal component analysis on them. Pretty much I wanted to take all the tracers that had a 50% or more detection rate, throw them all into a PCA and see how it would spread my data. And for the dry and wet season, it did group the urban environment compounds that were positively correlated with things like caffeine, 6-BPD, stormwater tracers, nitrite, and ammonia, which we have seen among the trends of the nutrient data and the chemical tracer data. And then on the left side, which is being influenced by these compounds here, we do see the nitrate, the sucralose, the carbamazepine influencing some of these more northern sites. So We still got to dig into this data more, but we are seeing some nice spread with the use of these combination of tracers. And there are differences in the wet and dry season. The lower urban environment sites tend to cluster more tightly together, where we have more spread between the northern sites in the wet season, potentially indicating stronger sources in the wet season compared to the dry season. And we can take this even a step further. I just wanted to plot just specifically the wastewater tracers, the sucralose, caffeine, and carbamazepine. Where we have sucralose and carbamazepine, I would consider this the legacy wastewater, like reclaimed wastewater treatment plant influence sites on the right, to where on the left would be the new wastewater, where it's got positive correlations with caffeine, ammonia, and nitrite. But again, so much to do with this data. Okay, so preliminary conclusions. So we did identify nitrogen hotspots throughout the river. We developed the tracer method where we do have effective tracers identified. Want to add some more in the future, but we did find correlations between those tracers and the nitrogen species. Overall, the total nitrogen levels were below the nutrient numeric criteria. There's areas to definitely dive deeper into in the wet season specifically for phase 2 of this study, but yeah. So next steps. So right now we have the Max Planck Institute for Chemistry in Germany who are conducting our isotopic analysis, our N15 and 14 ratio. So we're going to have that extra layer of data to help further hopefully tease out some of these sources. We actually got some additional funding, so our sampling for this project is going to end June 2025. And then I'm going to put this all into a publication, which I'd be happy to share with the group once that is ready, on the chemical tracers, nutrients, and isotopes. And pending funding for year 2, we're actually going to go into these hotspots that we identified, collect at different point and non-point source, and do some source apportionment. So we're going to revamp that sampling site and hopefully be able to calculate some loadings from those point and non-point sources, as well create a mixing model to visualize these different sources and their interactions and how they move throughout the river. And hopefully identify some areas for prioritize management efforts for some of these areas. And yeah, we have a nice list of collaborators, and each of them are super valuable as we meet periodically. They give advice, start to pick apart some of the data with me, and I can't thank them enough. And I'd be happy to answer any questions. Oh, am I muted? Oh.
You have a question in the chat from Maya, if you want to touch on that one. Can you see it?
I can ask it out loud for everybody too.
Yeah.
I mean, I think we currently do estimate nitrogen loading by major source categories. And so one of the hopes is that this work, might either validate that source apportionment or highlight some areas where we maybe need to refine our load estimation techniques. And so I'm wondering if you can weigh in on that. And then specifically, stormwater is the major source of nitrogen pollution. What are like some of the elements that are contributing to that stormwater load? Is your work able to speak to either of those questions?
Yeah, so it's our goal in phase 2 to go to the actual outfalls, the major outfalls of the river, calculate find the concentration of the nitrogen and nitrogen species, as well as the contaminants. And like you said, hopefully it will further confirm, your guys' robust data set, as well as provide specifics on like, in this area we have high concentrations of 6-ppd. It's more coming from areas around the urban roadway environment, compared to like adding some additional tracers for stormwater, like different like herbicides, pesticides that they would use to fertilize different areas and stuff like that. So really in phase 2, we're going to be revamping that sampling strategy, but also honing in on more tracers to hopefully determine and pinpoint exactly, you know, what it is, where it's coming from, where throughout the river is the hotspots for sure.
Okay, you have a question, which species might be most at risk from 6 PPD? We don't have many salmonids in this region.
Yep. Yeah. So that's a great question. And that's something a lot of people are chasing right now. So it's a super hot topic right now. 6 PPD, and it's actually 6 PPDQ that people are seeing that it has the most toxicity on things. I'm an analytical chemist, so I won't speak to toxicology things, but this actually started up in the Washington area where they'd had like a lot of coho salmon dying off, and that's where they found this compound. So now everyone's chasing it. So I'm sure there's going to be a lot of toxicity studies in the near future testing on different sensitive, aquatic organisms. So for sure.
I have a question if no one else has one. Thank you for coming back and talking to the TAC.
Yeah.
Hey, that rhymed. Maybe next year we'll have you back to follow up. So, yeah, with the updates to this, particularly the multivariate stuff, I'm starting to see a little bit more how this could be used in practice. Is really the goal of this analysis is like a fingerprinting source tracking method. And I'm curious, if and when this stuff does have utility for that purpose, have you been talking with some of the local monitoring groups here about sort of operationalizing some of the methods like Hillsborough County or City of Tampa? Because the laboratory equipment you showed has like great detection limits, and it seems to have some, or ramp, as Maya says. And I'm just curious, is it like practical cost wise to sort of add these items to sort of like, the routine methods to grab the water and sample for it?
Yep. So that's kind of exactly where we want to take this method. We want this to be something that can be used in a normal water quality where, I don't know, for the total nutrient suite, it costs like, anywhere from, our lab at FIU does it for like $110 or something like that. We want that to be comparable. So we are eventually going to open this up, this method up. And of course, once I publish this, other people who have the instrumentation can definitely adapt this method. And for sure, we want this to be transferable. We're going to be using it down in Miami-Dade County some more under Piero Gardinelli's direction, and for sure. Yep.
Cool, thanks.
Yep. And yeah, we want to do some more multivariate analysis. We were going to start, but since we got a little bit extra funding to keep us going till June, we're just going to wait till we have the whole data set to really dive in and tackle it some more, for sure.
All right. Going once, going twice. All right, I see no hands, I see no chats. Thank you very much, Cassidy.
Thank you all.
All right, up next is the Cedar Key Living Shoreline Performance Assessment by Savannah Berry with UF.
Hi, everybody. All right, let's get the screen share started.
You're good.
All right, wonderful. Well, I'm really happy to be invited to present to this group. And of course, I'm going to be talking about work we've done in Cedar Key, which is outside of your watershed, but hopefully the lessons learned and things that we have to show you will be relevant to your watershed. And I'm definitely eager to hear what folks think during the Q&A. So I'm Savannah Berry. I'm with University of Florida and Florida Sea Grant, and I'm based full-time in Cedar Key, and my appointment at the University of Florida is 100% extension, which means that I get to work with communities to apply science in their everyday lives and jobs, and a lot of my work has included helping the city of Cedar Key address some of their issues that they have using living shorelines. And so specifically for Cedar Key, coastal erosion has been a really big problem, and it's really accelerated since 2000, and that coincides with the acceleration of sea level rise rates as well as punctuation by several extreme events, as you all are well aware. But here is the same shoreline in the 1930s and 50s, where you can see nice fringing vegetated habitats, oyster reefs, low profile dunes, dry recreational beaches. And then that same shoreline present day has unfortunately been almost 100% armored with riprap and these like paved road aprons, as we call them, that are pretty commonly seen in Cedar Key. So that's the ground level view of the issues that the city's been facing. And then here's more of a bird's eye view of how this erosion has progressed over time. This is Daughtry Bayou, so it's to the west of the main area if you've been to Cedar Key and down on Dock Street where all the restaurants and bars are. This is somewhat to the west of that, and it is a fairly small embayment, but it is exposed to wave energy, especially from southwestern fronts and hurricanes. And you can see over time, these aerial images were taken at roughly the same tide stage, and we used to have a lot wider buffer for a lot of the residential areas and road infrastructure. And then in addition to that, road infrastructure has been added over time. For instance, in this 1961 image, we didn't used to have a road going down this corner, whereas that was built in the early 70s. And then that's when the riprap was installed. And over time, even as late as the mid 90s and early 2000s, we still had beaches and vegetated habitats along all of these areas, but then 2016 we start to see complete loss of those fringing buffer habitats and exposure directly to high tides of a lot of road infrastructure, sidewalks that have the tide lapping up against them. And so this is obviously a problem for the city, and it's created a lot of vulnerabilities, not just for road and sidewalks, but also people's homes, pool decks, utility lines, and things buried underneath the roads. So the city approached UF back in 2013 was actually the earliest discussion, but that has led to basically a decade of collaboration with city officials and property owners to try to soften some of these shorelines and restore or enhance historic fringing habitats using living shorelines. And so co-design has always been a really big underpinning of everything we've done in Cedar Key. We always have workshops with the community where they can contribute to the design, interact with consultants and contractors that we may hire. We've brought in a lot of grant funding to build these projects, and there has been some local investment, but for the most part, it's been external dollars. But you can see Cedar Key has one road in, and then it's just super exposed. So this is a list of all of the projects that we've completed or are working on with the city right now. And overall, these projects have served to soften more than 30% of this shoreline, and it has made it so there's very little direct armored infrastructure in contact with the water without at least some green buffer in front of it. So I'll tell you a little bit about 3 of the projects: the main section of Airport Road, Joe Rains Beach, and G Street, the part that's been completed so far. And I'll just step through a series of images here to give you an idea about what each of these projects consisted of. They each had somewhat different designs. As many of you know, living shorelines have to be designed to very site-specific factors, and Cedar Key's living shorelines were no exception to that. So in the top image is the pre-implementation state, and it's a little bit hard to tell because this is an overhead image, but there is a paved road apron similar to the one you saw on G Street that was being really severely undermined. We did have some existing low marsh vegetation and a little bit of uplands that were still vegetated, but all of these areas were retreating a lot, and the elevations were relatively low, again, with the tide basically coming right up to this hardened infrastructure. And then this is 3 years post-implementation. We built shore parallel reefs to serve as some offshore breakwaters. There's 16 reefs, and they're made out of pallet ball reef balls, which are 1,300 pounds each and are about 3 feet tall. And they were placed on the -2 foot contour to be right in the optimal growth zone for oysters. We also imported a lot of sand to this project to raise elevation so that the low marsh could be pushed waterward, and then we could install high marsh and dune vegetation behind in the higher elevation zones. And I'll talk a little bit more about that as I show you our vegetation data. But this was the basic design with these detached breakwater reefs and raising the entire elevation using sand and then planting into most of that area. And then here's G Street. This is the site that I was showing you the historical pictures of in the beginning of the talk. Again, we see this road apron, and the high tide would basically be right up to this sidewalk and before implementation. And then after implementation, you can see that this is more of a marsh with sill design where we have an outer sill in this patch of marsh. And we also installed a couple of shore perpendicular oyster reefs. They're very low profile. They don't interrupt the hydrology like an emergent groin would. We call them point bars so that we can avoid the terminology groin. And these do actually mimic natural features that we see in Cedar Key. If we were to zoom out on this image and look north along the shoreline, we would see these shore perpendicular oyster point bars. So often when we're making our designs, we're mimicking nearby natural features that seem to be keeping the shoreline stable. So that's the G Street project. And then this is the Joe Raines Beach project. This is the first project we did in Cedar Key, so it's the most mature of all of them. And this one was built in front of a failing bulkhead up here. Was a city park. And then there was also some private property involved in this project. And the bulkhead was being undermined by high tides. And then also there was a historic canal here that had been filled in by all the sand that eroded from this site and was deposited down here. So this project was able to make use of local sand through the dredging that pumped the sand back up. And instead of just leaving it as sand, it was planted into for most of it. We did leave a small portion that was unplanted to serve as a pocket beach, but this included low marsh and high marsh. So this was another marsh with sill design, but it was in front of a vertical bulkhead instead of a road apron. So you're probably thinking as I'm going through this, wow, all of these living shorelines are built in front of armored infrastructure. So yes, these are technically living shoreline retrofits. We did not remove any of the armoring that was present at these sites, mostly because of cost and because there was no space behind the armoring due to road infrastructure or very narrow setbacks of city infrastructure and properties. So there's no way that we could have removed the armoring and then regraded the shoreline, for instance. So we had to work around the existing armoring. And as you'll see, it presented some challenges, but we have definitely seen quite a bit of success through this approach. But I'm not going to go into this data, but in the paper, you can look up, we did analyze, time horizons for when these projects would be squeezed out by sea level rise, which is certainly a reality of doing retrofits instead of true living shorelines. All right. So next I'm going to show you some data about the performance of vegetation, and all of these slides are going to be formatted the same. The data that's shown in the line plots is exactly the same data that's shown in these aerial images. But I think the aerial images are a little bit easier to interpret, but I included the lines because some people like to look at those. And all of the legends are going to be the same on this slide and the next two. The blue is dune vegetated community, the orange is high marsh community, and the green is low marsh, and brown is mangrove. And then the lighter colors indicate that the vegetation type is present but not dense, and the darker polygons are where that vegetation is dense, which is basically you could consider fully established. We're starting with Airport Road, and then also the last two images on all of these slides are going to be directly before Hurricane Idalia and directly after. And Hurricane Idalia, at the time this data was collected, was the most extreme storm surge event that we had experienced in Cedar Key. Unfortunately, it was outdone the very next year by Hurricane Helene. And all in all, these projects have actually been hit by more than 7 tropical storms and hurricanes since they were installed. But Hurricane Idalia was the worst one and the one that we had the best data for. So that's why we focus on that one for this paper. But overall, we generally have had an increase in both diversity and vegetative cover at Airport Road. and we were getting really nice establishment, especially in that upland transitional community. But unfortunately, that was the community that took the heaviest hit during the Hurricane Idalia. And that is pretty much because this is right where the wave action is. This elevation is getting churned up and battered by wave action because the water levels are so much higher. And so unfortunately, we did see certain areas of that upland transitional community be lost completely. And that's because the riprap that was underlying this area of the road, it allowed the vegetation to be completely scoured out and lost, and even the sand underlying was lost. So that was a big takeaway from this project, and was that upland transitional community is really important to try to establish the full spectrum of an intact shoreline. But it's really hard to get it established if you're going to keep getting hit by hurricanes, as Cedar Key has. So a lot of living shorelines are low marsh with sill, and they don't really contemplate or attempt to restore higher elevation vegetated zones, but we thought it was important to try to restore all of that cross section. So now here's the similar data for G Street, and you can see this is pre-implementation up here. There was some existing upland transitional. This is mostly a Christmasberry shrub patch, and then after we installed, you can see that we had an enhancement of all 3 vegetation zones again. Initially, we had a lot of trouble getting the high marsh to establish on top of that concrete apron. It's not so easy to get plants to grow in only 4 to 6 inches of sand on top of concrete. So we had to do some repair of that high marsh, but we were eventually able to get it to establish, and it was able to survive and persist through Hurricane Idalia. And interestingly here at G Street, we didn't see a collapse of the upland transitional and high marsh communities like we did over on Airport Road. And we think that it's mostly because this was more based on woody vegetation like the shrubs, instead of being a purely grass like sea oats and panic grass, which is what the Airport Road site did. So we think that's an important lesson learned for projects that are restoring the entire cross section from Oyster Reef all the way to uplands, to consider maybe looking at more woody plant selection. That's a new idea that we've had, but anecdotally on the ground, we've seen it persist much better through these extreme events. And then lastly, this is the Joe Rains project. We had the longest time series of data for this one. And you can see that we got the best establishment here at this project overall. This is the only project that didn't get hit by a hurricane within the first 2 years of installation. G Street and Airport Road were both hit directly by Tropical Storm Eta, and then the next year by Hurricane Elsa. And the first time that it got hit by a tropical event was within 2 weeks of them being installed. So we definitely have had some limitations of G and Airport Road reaching their full potential because of just really early bad luck of getting hit before they had a chance to establish. Whereas at Joe Rains, we've seen almost no impact of hurricanes whatsoever to the vegetation because it had time to establish before it was challenged by any type of surge or significant wind energy event. One of the other things to note here at Joe Rains is that we've seen really significant encroachment of mangroves and starting to outcompete even in the low marsh, this sort of upper edge you see down here, all this brown. These are all mangroves that have naturally recruited into what we just planted as an herbaceous marsh system. And this is not really maybe that relevant to Tampa Bay because you all already have a lot of mangrove dominance. But here in Cedar Key, this is really interesting to see the evolution and how mangroves have started to become dominant over marshes and outcompete them. And this is just a fully natural process that has started to occur here. And we've also seen natural recruitment of upland species. We did not install any upland transitional at this site. We only did high marsh, but we've seen that as it's captured sand, it's actually built up even to upland elevations and had natural recruitment. So Joe Rains is doing well, and we hope to see similar maturity in the other 2 projects as they age, even though they've been completely battered by hurricanes. Okay, so speaking of hurricanes, I know data's all great and everything, but I think a lot of people get more from looking at pre-post photos and photos from on the ground. So next, I'm just going to step you through several series of photos just so that you can get an idea of what this looks like on the ground. And to give you an idea of how extreme Hurricane Idalia was, and of course Helene was worse than this. This is our living shoreline down here at the end of the road. This is the G Street living shoreline. So, and that's 6 weeks after Idalia, and you can see there's still lots of grass. And we didn't do any repairs after the hurricane at this project, so it's there and happy, even though it was covered up by huge amounts of water during the hurricane. So let's look at another time series. We're going to go all the way back to 2022. And again, like I said, this is the high marsh area that we had an initial failure and we had to come back and repair it. And this was April 2023, which was only 6 months, give or take, before it was hit by Hurricane Idalia. But the high marsh grass was able to get enough of a foothold after we installed these speed bumps, as we call them, along the concrete to interrupt the wave energy that would just propagate along this flat armored surface. So that was an intervention, kind of an adaptive management, if you will, of how to get this established. And it did last through Idalia, and it showed really nice recovery 6 weeks after Idalia. As you can see, we've got a nice high marsh coverage there. And I keep on mentioning Helene, and I haven't even talked about Debbie, which was another hurricane that hit earlier before Helene and Cedar Key. But I don't have data from the aerial images to show for this, but I do have on the ground photos to show, like, you know, after Idalia, this is the next summer. We had awesome recovery. This is all self-repair of this shoreline. This is nothing that we came in and replanted. And then we had post Debbie, you almost can't even tell that this site was hit by anything. And then similarly, 1 month post Hurricane Debbie, just before Hurricane Helene, it looked similar. And then Helene, of course, deposited a lot of debris, but we still had a lot of vegetation there and we didn't have any appreciable losses here at G Street. Of course, the sand, this area back here was meant to be a beach and the sand got washed away, but it was important. I mentioned co-design was a really important part of these projects, and for the community members, trying to keep a sandy beach in a small area was really important, even though, you know, we communicated with them that that was going to be very hard to maintain without continual addition of sand. And so that's the main impacts we've really seen at G Street have been the loss of the sandy beach, whereas the vegetation has held on really strong. Now moving over to Airport Road. And these are pre and post Idalia photos. So the top set is pre-Idalia. You can see this is that upland transitional area that we were having really nice, fast establishment, even with some of the sand being lost from that initial set of hurricanes that hit them. We still had a lot of plants trying to make it and grow with the sand that was remnant within these rocks. But then after Idalia, you can see the low and the high marsh did okay. There's definitely some signs of stress and a little bit of sand deposited on top of the plants, which is no problem. They can handle that. But this upland transitional area was completely collapsed due to the storm surge just washing out this area. And then this is just another part of the project, and it shows the same story with the high and the low marsh persisting just fine through the storm surge event, but this upland transitional zone being completely lost. Further down the shoreline where we didn't have the riprap, we saw less of a total collapse of the upland transitional. You could see up here, we still have some remnant vegetation. A lot of the above ground and leaf biomass was lost, but there were still remaining rhizomes. And we actually started to see in this area that a lot of those rhizomes were able to put out new above ground biomass and shoots and try to recover and self-repair in this upland transitional. So that was after Idalia. But then this is the 2024 set of hurricanes, which again, I don't have data for, but we have good photos to show you this upland transitional part. We did have this armored road apron that was a problem, but we didn't have the riprap. And we started to see lots of self-recovery, but then Hurricane Debbie again made the upland transitional zone lose a lot of its leaf biomass. It was able to come back pretty quickly from that event, and then Hurricane Helene did the same thing to it. And unfortunately, even though we have seen some weak recovery, we've seen a much less strong recovery of this upland transitional vegetation zone because it's just been completely, it's lost its above ground biomass now 3 times in the last 2 growing seasons. And that's just, there is a limit to what these projects can handle. Of course, there's still a lot more vegetation there, even after all these extreme events, than there was before we did the project. But, I think we have to be realistic about how many times we can expect these projects to repair themselves. So coming in and doing some supplementary planting and sand placement is probably going to be part of the long-term sustainability of these projects. But overall, we're really impressed with how they've handled what they've been confronted with, and they've exceeded our expectations in many ways. All right, so now I want to talk a little bit about some wave attenuation data that we've been able to collect. So all of that was mostly about vegetation performance. And we have been working with Mississippi State University that has developed these low-cost wave gauges that have been validated against the more expensive industry standard wave gauges. And so we've been able to construct quite a few of these and deploy them during both typical conditions and hurricane conditions to measure wave attenuation around these living shoreline sites. And the sort of overall takeaway is that in typical conditions, we measured between 33% and 79% reduction in wave energy, and then up to 28% reduction in hurricane conditions. And this was definitely consistently outperforming what we were able to measure at armored shorelines. And during typical conditions, we often measured armored shorelines amplifying wave energy due to reflection of that wave energy back out into the coastal environment. And then during an extreme event, even though we didn't see wave amplification from armored shorelines, they definitely underperformed compared to living shorelines. So I'll show you some of that data. This is just for maximum incident wave power. If you look at the paper, you'll see a lot of different measurements related to wave height and other things. So I'm just going to show you a subset of the data, but this is how we collected this data. We deployed paired wave gauges, and this is during Hurricane Idalia. I'm not going to show you any of the typical conditions numbers until the end, but during Hurricane Idalia, we had these outer wave gauges that would represent the incoming wave energy, and these inner wave gauges that would represent the wave energy after it had interacted with either the living shoreline elements, or in the case of this site over here, which was our non-reef reference, we just measured wave difference from the inner and outer, and any difference that you see would be related just to the shoaling of the shoreline, because there was obviously no reef here. So at Airport Road, we only measured wave attenuation related to the reef ball reefs. If we had put our inshore wave gauges a little bit higher in the shoreline profile, we would have been able to measure what was going on related to the additional vegetation roughness and shoaling that was created. But for the purposes of this study, we only looked at the reefs, and you can see the yellow numbers are the percent reduction between the outer and inner gauge. And we were somewhere around 11.5 to up to 15% wave energy reduction during Hurricane Idalia from the reefs, whereas at the non-reef site, it was about 5.5%. So about 10% more was related to the reefs. And then this is the similar data from G Street. And at G Street, we had an armored reference, which is the one on the far right. And we had 2 gauges deployed within the living shoreline. So this actually does capture some of the effect of the vegetation. And then we had a natural reference over here, which was a vegetated shoreline that was just to the north of the living shoreline site. And the main take home here is that the wave attenuation numbers that we measured during Hurricane Idalia for the living shoreline were on par, or slightly less than what we measured at the natural reference site, but far exceeding what we measured at the armored site, which was a riprap site. So only 2.8% wave reduction from the armored shoreline. All right. So the wave attenuation data from the hurricane really surprised us, because honestly, we were expecting it to be no effect. because as the water gets deeper and deeper, those living shoreline elements are getting buried under that, they're getting submerged more and more. And there's really only an opportunity to reduce wave energy when the living shoreline elements can interact with where the waves are in the water column. And so in our minds, we were thinking as the water gets deeper, the living shoreline's really not going to have an opportunity to do anything to wave energy because it's just too deep in the water column to do so. But we didn't account for wave period when we were making that prediction. So it's nice that we actually have this data. So what you're looking at on this slide is data from G Street and Airport Road, and it's energy reduction on the y-axis versus water depth on the x-axis. And the dotted lines here are if you just looked at the blue points, which is basically small period waves, which is the typical conditions. So we have mostly wind waves here in Cedar Key. At least in this area, there's not as many boat wakes. So it's mostly wind waves, and they're very short period waves that don't go very far down into the water column during our typical conditions. And so those are the blue. And if you just look at the blue points and you kind of extrapolate a trend, an energy reduction with water depth based on the blue points, you would expect the effect of the living shoreline to go to zero sooner than what we actually observed during the extreme event. So at G Street, we would have expected to see zero effect of the living shoreline at a water depth of about 2.75 NAVD, whereas at Airport Road, we would expect to see it drop off even sooner at about 1.75 NAVD to zero. Whereas during the extreme event, when we had higher wave periods, we actually saw what we were calling a performance bump, and we actually saw that the living shorelines were able to attenuate more energy for higher water depths than would be expected based on typical condition measurements. And so that's basically because when the wave periods get longer, the waves dip deeper down into the water column, and then that means that the living shoreline actually has an opportunity to interact with and reduce that wave energy, whereas it wouldn't if the wave periods were not longer. So I hope that makes sense. I feel like I was waving my hands a lot during that explanation, but it speaks to the importance of trying to collect measurements across the spectrum of conditions that these shorelines are going to experience. All right, and I know I'm running a little short on time, but I just want to briefly mention this is a different study about perceptions of different shoreline management strategies, and it was a statewide survey that we did of shoreline property owners, and this is published, so I'm going to blow through it pretty quickly, but if you want to check it out, it is definitely relevant to Tampa Bay because we focused on 3 regions of Florida: Indian River Lagoon, Panhandle, and Tampa Bay. And we actually found that perceptions were really not different across these regions. So region kind of disappeared from our analysis, but it was interesting to see that across the state, perceptions of different shoreline management strategies were similar. So overall, we basically asked people about 4 different shoreline types: armored, hybrid, living shoreline, and natural. And we asked them to give these shorelines all kinds of different ratings and ask about what would drive their decision-making for selecting different types and those sort of things. So some of the highlights for this was that homeowners generally rated natural and living shorelines lower for values that for functions that they valued the most, like protection scores. And interestingly, they also ranked cost as a really important decision-making factor. But most people in the survey had armored shorelines. But people that did have living shorelines reported that they were about 8.5 times cheaper to install and 2.7 times cheaper to maintain than what armored shoreline owners reported. And another thing that I thought was very unfortunate, but perhaps presents an opportunity for education and outreach, is that homeowners generally reported using very few sources of information when making their decision about what type of shoreline management strategy to install. The average was, I think, 1.4 sources of information. So I think that living shorelines aren't getting a shot because people maybe just aren't learning about them. Overall, we found that people, no matter what type of shoreline they had, people that live in Florida that own waterfront property were generally pretty satisfied with their property. So the overall average satisfaction with their shoreline was 7.5 out of 10. but that jumped to 8 out of 10 if people's shoreline was either stable or had gained since they had owned it, whereas people that had an eroding shoreline or were unsure were more like 6.4. And interestingly, people with natural shorelines had the highest overall satisfaction of any group. And so that was also kind of a bright spot that if living shorelines can mimic natural shorelines, then people will probably be most satisfied overall with those. I'm not going to go through this whole figure. The main take home was from this part of the analysis was that neighboring shoreline type has a really strong influence on the type of shoreline that their neighbors will in turn have. So folks that have armored shorelines are much more likely to live next to people with armored shorelines, and people with natural shorelines are much more likely to live next to people with natural shorelines. And that also applied for hybrid and living shorelines. So there is a domino effect, and this has been shown in other states that have looked at this, that people tend to kind of copy their neighbors, even if they say in the survey that they don't do that. There's evidence that they are doing that. And then the last thing I want to say is that there was overall really limited understanding of shoreline connectivity in the survey. A lot of respondents totally misunderstood how their shoreline could affect their neighbors and vice versa. So for instance, people with natural shorelines gave their shoreline a benefit rating of the lowest overall average of any group. So for this part of the analysis, we asked people to rate how their shoreline would affect their neighbor from very beneficial to very harmful. And people who had natural shorelines gave their shoreline only a mean rating of 2.7, which is basically neutral. Whereas people that had non-natural shorelines, hybrid, seawalls, those sorts of things, gave their shoreline a higher rating. And then actually the highest rating of any group that we measured were people that had very large seawalls. They thought that their shoreline was benefiting their neighbors the most. So that was definitely unfortunate because we know that armored infrastructure can cause harm, direct harm, not benefit to neighboring properties. And so there, again, another opportunity for education of our folks. And so there seemed to be maybe that hybrid shorelines might be a middle ground for getting greater adoption of some nature-based approaches, at least for people that may be hesitant based on negative perceptions of natural infrastructure. All right, and I just want to acknowledge all of our funding that we had. Like I said, this is almost a decade of effort now for all of this. And so I'm here on behalf of a very large team of collaborators and volunteers and agencies that have supported our work over the years. So thank you all again for having me. And if there's time, I'd be happy to take questions.
All right, it looks like Stacy had a question. When was the survey conducted in relation to the big storms in 2024?
Yeah, the survey was actually done in 2019. So it was done quite a while ago. It just took us a while to get it out.
Randy, you have your hand up?
Yeah,
Yes.
on the mechanism of wave attenuation, is it when the water gets higher, is it more the waves dipping lower, or is it because the water column under those waves is in circular eddies, and maybe the structure is interfering with the bottom of the eddies of the waves and the vertical movement of it?
Yeah, I'm not sure. I'm definitely not a coastal engineer or anything like that, so I'm not sure the exact mechanism. We hypothesized that it was wave period, and I don't know if wave period would interact with how far those eddies would extend down into the water column. So yeah, I'm really not the best person to answer that, but I think either way, it has to do with the fact that the waves are larger.
Mm-hmm. Thanks.
All right, and then Randy, there was a Randy Edwards as well. Sorry about that. Should have used the last name. You had a question or comment.
Yes. First of all, to the IFAS people, I have to say, go Gators. But beyond that, I have to say that when I see any project like yours, and I know this is not your area of expertise and probably not your decision making, but when I see reef balls being used either for habitat enhancement or for breakwaters and so forth, I have to say that it really triggers me because in my view, my experience, because I study fish habitats, I did projects on artificial reef habitats, and reef balls, by every measure I can think of, are probably the least effective structures to use for those habitats and for that purpose. For example, they provide the minimum amount of surface area. A sphere, if you take a drop of water, you make it into a sphere, it minimizes its surface area. Surface area provides habitat. It provides almost no heterogeneity of structural habitat. Big half semisphere with few holes in it provides very little habitat for the fish species that you are hoping to enhance as you're doing these kinds of things. I used to sit when I was at the USF St. Pete. I used to sit on the seawall where they have reef balls along the shoreline behind the library, and there was very little use of those. There are many designs that could be much more effective to provide much better fish habitat and probably better wave attenuation than those hemispheres. So I just urge you to talk to your colleagues in these kind of programs and projects and try to figure out some habitat friendly type of structures to use and not just go to reef balls. They've been pitched widely, but I would say that they are the least effective type of structure you can use in these kind of projects.
Well, I just have to respectfully disagree with you. I didn't show the data, but we've had massive and impressive oyster recruitment on the reef balls, which of course increases the surface area and the complexity quite a bit more. We also had private property owners that were involved in permitting and designing these projects, and we showed them a range of material options. There were, limestone rock reefs, we had wave attenuation devices, we had several other types of options they could have selected from, and the strong, clear preference for what those people want to look at every day out of their waterfront view was reef balls. So, there's lots of factors that go into material choices and types, and we didn't design this project as a hurricane mitigation or a wave attenuation project. It was a habitat enhancement project. We built it using dollars that were meant for habitat restoration. And so our oyster restoration goals for the project have been achieved really well by reef balls. But I mean, if they're not your taste, that's certainly your prerogative to choose different materials.
It's not a matter of taste. It's a matter of habitat for other fishes, particularly.
Okay,
Sure,
well,
for
our
oysters.
goal wasn't fish. Our goal was oysters.
Well, that's the
and wave
typical goal, and
mitigation.
that includes NOAA's emphasis on living shorelines.
Okay.
Ed?
Thanks, Savannah. Great presentation, great research around this topic area. I had kind of two things I just wanted to mention and get your feedback on. In relation to the performance of those living shoreline installations in Cedar Key, what were the perceptions of the residents in terms of their view of how well they performed versus other areas in and around the town?
Yeah, definitely. So I'd say that Joe Rains being the first project we built, and it survived really well through like Hurricane Michael, even though that didn't hit Cedar Key directly. We had 5 feet of surge from that event, and they saw it was pretty much untouched from that. It actually captured sand during Hurricane Michael, and it was the first time the city hadn't had massive repairs to that seawall after overtopping. And so that really won a lot of people over. And then the fact that we involved the community in the design from day one, and we hired external facilitators, we weren't necessarily trying to push folks to any one specific design or outcome. So I think that genuine engagement also really helped, but we have not been without our objectors. Certainly our plans for the G Street project were actually to do the entire shoreline. If you were looking closely, you would have noticed there was a portion of G Street, that apron that we sort of stopped 75% of the way down. And that was not the original plan. Our original plan was to do the entire shoreline to the corner. But we had some folks that had a dock, and they were really concerned that the living shoreline would cause sedimentation around their dock, and we just couldn't come to an agreement. We didn't have funding to run a full hydrodynamic model to sort of allay their concerns, and we weren't able to come to an agreement. And so we had to sort of build the project in a way that it wouldn't intersect with their property. So certainly we've had objections. Also, mangroves have been a really big deal here in Cedar Key, and we did even reach a point where we had to build in mangrove management into the permit in order to make the property owners feel comfortable moving forward because of the obviously fear that mangroves would recruit into the marsh. Everyone was fine with marsh, but the fear of having mangroves come in and be a real management hassle for them. We had to surmount that during the permitting process, and DEP was awesome working with us on that. So yeah, it's definitely not all roses, and we have won more and more people over with the years as they see them continually persist and persist and persist. So that's been a benefit, but there's also a lot of turnover of homeownership in Cedar Key, and so we are constantly getting new folks that are buying properties that we've either tried to work at before, and then we're getting calls like, hey, why were we left out of this project? And it's sort of like, oh, well, the person that lived there before told us to go away. So yeah, it's been interesting, but it's a really important part of living shorelines is that social aspect. So yeah, thank you for asking that question.
Yeah, that kind of relates to the second comment I had in relation to your observations, seeing the mangroves recruit in some of the salt marsh. That's some of the lessons learned from some of our early restoration projects. And I dropped a link into our Habitat Master Plan Restorations Best Management Practices Manual that kind of highlights that observation to some of our early restoration and this idea of if you plant the marsh, you're most likely going to get mangrove habitat now as our coastal habitats are changing, and I'm sure Cedar Keys are going to continue to change. So that sort of next step of perceptions of folks liking marsh, but not necessarily mangrove recruitment. And you kind of addressed it in your earlier comment, but that's an interesting dynamic to deal with as the coast continues to change, especially up there.
Yeah, well, thank you for sharing that resource. I clicked on it and I'll definitely have a look at that. So thank you. I saw a question in the chat about reef balls and affecting turtle nesting. We don't have sea turtle nesting in Cedar Key, but I know that this has been extensively looked at for reef balls, and I'm sure they wouldn't be allowed to continue to be used in Florida if there was a problem. There's an escape hole at the top that is supposed to allow turtles, if they can fit into the side holes, that they can also fit out of the top hole and escape. So I believe that's been extensively looked at, but I'm not familiar exactly with the data. But yeah. Hi, Mark. Do you recognize some of those projects?
Yeah, when I actually had the ability to do field travel, since I don't have a budget, I'd like to go out and ground truth those at some point in the future. Dr. Berry, great discussion. Thank you as always for your presentation. Just some advice and a question for you, just with regard to siting these structures. We strongly advocate, in fact, the Corps of Engineers nationwide permit 54, I believe, requires that living shoreline structures not be placed in areas of submerged aquatic vegetation or seagrass habitat. And that's typically not the case. I do work with a number of consulting agencies throughout the Gulf Coast of Florida just to try and avoid and minimize any potential impacts to seagrass habitats. But my quick question for you was, I found it interesting with the monitoring of the additional habitats on the backside. Have you seen any recruitment of SAVs or seagrasses in these areas that were, formerly induced shallow water wave, high energy type areas? And then you put in these, in this case, oyster domes to attenuate the wave action. Any recruitment of grasses in your experience there, Dr. Berry?
No, and my background is in seagrass, so I love seagrass. But we actually don't ever see seagrasses in this bay of Cedar Key. I know that there is a layer out there that says that we might have some sparse halodouli in certain parts, but I have never seen living seagrass at all in Daughtry Bayou, and we haven't seen any recruitment of seagrass. And the water's really, as you know, super tannic, super high particulate load in Cedar Key. So I don't think it's a very good environment for them. A lot of these are like mud flats that turn over a lot with wave energy and also with just poor light conditions. So yeah, we've seen tons of oyster recruitment, tons of recruitment of other native plants that we did not initially install in the marsh side, but no SAV.
Well, I'm wondering, with the improved water quality through the oyster restoration, right? I'm wondering if maybe we would get a reduction or an increase in light transvicity where there could be recruitment of SAV. Because you're right, there's that band. I dropped it in the chat there, the FWC mapping tool that we use quite a bit. Potential seed source or source for rhizomes. As they break off, maybe they'll float in on the backside and we might get some SAV habitat as well. But enough about SAV. I know it's important. So great discussion and good to see you.
Yeah,
Take
you
care.
too. And I don't think, honestly, there's been people that have tried to measure improvements in water quality based on the aquaculture industry here, because it's like our little oyster restoration is nothing compared to the amount of clams that the Cedar Key farmers are out there farming. And it's basically not even measurable from that amount of biomass. So there's a lot of nutrients in this water, and the Swanee is pumping out a lot, and there's a lot of water movement, a very low residence time. So I'm sure that they're filtering a lot, but they're not having the opportunity to reduce particulate load because there's just so much new water and that's at least what I think. I just don't think that, I think it's one thing that protects us from having really bad algal blooms is that we do have low water residence time in this area. We don't have these restricted embayments where water just sits. And so, yes, we have a lot of filter feeders, but the load that they would have to filter in order to reduce it enough noticeably, given the amount of water turnover, I just think the math isn't there for that.
Yeah, I agree with that. That's a good point. Good observation. Thanks again.
All right. Any further questions or comments for Savannah? All right, seeing none, do any of the TAC members or have anything they want to bring up or something else as you thought of something else for one of the speakers? I'm not seeing any hands. I'm not seeing anything in the chat. So Marcus, I will turn the announcements over to you.
So we got a few announcements, and I think I'll turn it over first to I think Maya. If you want to handle this first one, I could do the last two.
The first one is just a memo that was drafted by Dr. Randy Edwards, sort of documenting the conditions that he has observed in the Braden River. This is a follow-up to comments that he provided at the last TAC meeting. And he asked that we share this document with you. So I'm sharing it here at his request.
Randy, I don't know if there's anything you want to say about that.
Probably not. That document is so long and so detailed, it probably speaks for itself. So thank you for providing it to people so that they can consider what's going on here. Thank you.
Yeah, and I'll just offer that, we are working with partners, as we discussed at the January meeting from FWC, to analyze the data that are available to start to look at this issue that's been brought to our attention. And so we'll be bringing this back to the TAC at a future meeting so that you all can, as part of your roles and responsibilities as being on the TAC, you can look at that first analysis of the existing data and provide counsel on, what an appropriate course of action may or may not be based on that look. And so just a reminder that these aren't passive meetings where you just come and you listen to presentations, but we're also asking of you to help weigh in and provide guidance on the direction of the science needed to manage the estuary. And so stay tuned for that.
Yeah, and that discussion is possibly going to happen at the next TAC meeting in June, depending on how quickly the data can be analyzed. So we'll definitely share information beforehand or sort of set the expectation that we're hoping to have sort of a constructive discussion about the outcome of that in terms of what might be done or concluded from those data.
And Marcus, before you go on to the next ones, which were announcements that I dropped in there too, I just also wanted to bring to the TAC's attention that we're probably due for a presentation on the Old Tampa Bay Assimilative Capacity Assessment Project. There's a number of recommendations for ways that we might think about new indicators that we need to be tracking, or ways that we need to think about different targets or thresholds related to water quality and chlorophyll production in the Old Tampa Bay segment. And again, these are not things that the estuary program just decides and then pushes down to you all. We involve you in the decision-making process along the way. Many of these decisions are sort of within the purview of the Tampa Bay Nitrogen Management Consortium, but there are several new proposed indicators and targets that would really be up to you all as the TAC to consider, weigh in on, and make a recommendation to our management board. And so we're getting to the point where we're ready to sort of involve you more thoroughly in those conversations. And again, we'll be looking for your feedback about how we ought to manage the Old Tampa Bay segment into the future to see the kinds of outcomes that we'd like, that we hope for as a region in the Old Tampa Bay segment. So stay tuned for that as well.
Yeah, and that will very possibly happen in June as well. All right, I can do these next two items. The second one is you've probably seen the email about this. We send it out to multiple lists that we curate, but there's a support letter being circulated that's basically asking for your support for the National Estuary Program as a whole, not just us, but how you view the value of that program to our nation and the important estuaries that are around. So this is a letter of support that's been drafted by the broader parent organization of the Tampa Bay Estuary Program, so the Association of National Estuary Programs, that just asks you to express your support. Basically, provide your name, affiliation, where you're from, and that will be when that's, well, by the 31st. So please sign that. And when that's been signed and that's been closed, that will be sent to Congress to basically express broad support for this program. And of course, as we all know, that's, this is a critical time for that support among all the other things that are happening. So this is just one way that you can, express how valuable we are to you as a community. And then the last announcement is there's going to be Boyd Hill Trek and Chat happening on the 29th. There's a link on the agenda to register for that. Yeah, it'll be a Trek and Chat, and I think it'll be a great opportunity to get out there and experience Boyd Hill if you've not been out there. But definitely sign up for that if you have an interest. And yeah, contact Sheila if you have any specific questions about that. But
actually, you can contact me because I'm leading that one. And I just, for any of you who might have been receiving complaints or communication from concerned citizens, this Trek and Chat will really focus on exposing people to the ideas about natural channel design. I know I've heard from many residents in different communities throughout the watershed that this is a topic of concern. And so if you've heard from some of those people and you have residents that might be primed or interested in this messaging, I'd encourage you to share this registration link with them, because we will be talking about sort of ditch management and stream restoration techniques. And that's really the audience that I'm hoping to get out and talk. And like I said, I'll be leading that hike, but we'll also have our partners from the Water Management District there to help answer questions too. I promise I'll make it fun.
All right, thanks for that added context. That's it for announcements. If anybody else has anything to add, feel free to speak up now. Otherwise, we will adjourn the meeting, and the next one will be June 4th. So appreciate everyone's attention today.
And this
thank
is
you to our
Stacey
speakers.
Day. Sorry, I put, we are going to be installing some more living shoreline. It's part of our seawall enhancement out at Philippi Park. And I put that in the chat. It's April 5th and April 19th in the morning. And I can share the link also for sign up. Oops. Thanks.
Thank you, Stacey.
And if you saw Randy dropped a message in the chat regarding the physical move of their offices. So make sure you read that so that you know how to get them.
We will update our contact as well for that.
All right, if there's nothing else to discuss, our next meeting is June 4th. More to come on venue, in-person, virtual combo. We'll see what the presentations are, and that kind of our guest speakers kind of dictate, guide, how we host the meeting. So with that, we'll adjourn the meeting and have a great week.